Greetings, everyone, and welcome to Tipping Points episode number 25, where we're asking key questions about how to balance conservation and development. This webinar series is an initiative of Oppenheimer Generations Research and Conservation, and these webinars usually take place on the last Thursday of the month. So here we are at the end of August already at 1 p.m. I'm Robert Inglis, the Director of Jive Media Africa, Science Communication Partner to OGRC, the Oppenheimer Generations Research and Conservation. And I'm coming to you from the Jive Hub in KwaZulu-Natal, where about 10 or 15 kilometers away, it has already started to snow. So hopefully we'll be playing in the snow before too long. But the focus of today's webinar is on the JWO top 3 finalists. Congratulations, you're going to meet those 3 finalists today. We are going to be discussing their research proposals and what inspired them to pursue this research. Now, the JWO research grant, named for Jennifer Ward Oppenheimer, seeks to support innovative African-led research that will contribute to the advancement of environmental and allied sciences and address relevant real-world issues of current and future importance. The winning applicant for 2024 will actually be awarded $150,000 to pursue their research. And we're going to be finding out from them today about why their research is so important and so deserving of that grant. This year's winner will be announced at the annual Oppenheimer Research Conference, which takes place from the 9th to the 11th of October in Johannesburg. So really thrilled to be with you here and looking forward to productive and insightful discussion. This session will be recorded. It is being recorded, and it'll be available on the OGRC YouTube channel. This is also where you can catch up on any tipping points that you've missed, 25 in all by the end of this one. I really encourage you to sign up for the OGRC quarterly newsletter, where updates will be sent out, and you'll find those links in the chat. And then stay tuned to our social media channels for more insight around this. We're thrilled to have such a great turnout today. Please do continue to introduce yourselves in the chat. I gather the CBC is here in full force. There are another 15 people joining in one room from CBC. Don't hesitate to use the chat box to ask any questions of the panelists. This is your chance to meet these leading thinkers and researchers. So today I'm really pleased to introduce you to the JWO top 3 finalists for 2024. And I'm going to start with Dr. Nompumelelo Baso. Dr. Baso obtained her PhD in botany at Rhodes University. She's now a postdoctoral fellow at the South African Institute for Aquatic Biodiversity. And in relation to her research, she says that whether it's identifying aquatic macroinvertebrates or clarifying complex science concepts, she thrives on making science accessible. So hopefully that's what she'll be doing for us today. When she's not immersed in research, she'll be helping others to make sense of their data, or in the garden with her inquisitive toddler. So welcome to you, Dr. Baso. She adds her own secret pleasure is she's obsessed with RStudio programming. Cracking a code that's been refusing to run is the best feeling. Welcome. Our second JWO finalist is Dr. Shannon Conradie. Dr. Conradie is a postdoctoral research fellow at UCT, the University of Cape Town. She obtained her PhD from the University of Pretoria with a thesis entitled Novel Approaches Integrating Physiology and Behavior into process-based models of the impacts of climate change on desert birds. So at least some of us will understand at least some of those words, but we're hoping that by the end of this discussion, we all understand at least the concepts involved, because it's the details that are really hot for Dr. Conradie. Global warming is bearing down on all species, but Dr. Conradie wants a better understanding of thermal niches and to be able to predict wildlife species' thermal vulnerability under various combinations of global change scenarios across land and water in Africa. Welcome, Dr. Conradie. Have we got Dr. Conradie spotlighted? Dr. Steve Baleba is our third JWO research grant finalist. He holds a PhD in entomology from the University of Pretoria. The bees, the butterflies, hoverflies, and other pollinators all need a hand to keep our crops healthy and growing. And Dr. Steve Baleba wants to restore their habitats and create safer farming practices that don't rely on harmful pesticides. To meet Steve personally, he enjoys watching manga cartoons with his daughter, and he's a big fan of amapiano music. That is why his overall goal is to ensure that we can grow enough food while also protecting the natural environment and supporting the people who rely on these ecosystems for their livelihoods. So really thrilled to meet you all. First, from my side, a hearty congratulations to the three of you for making it this far in this competition. And of course, while there'll only be one funded researcher, we absolutely expect that your careers will be, or your research aspirations will be supported by this platform that the OGRC is creating. So we're going to first hear from Nompumelelo Baso. I'd like to hand over to each of the speakers for a 5 to 7 minute presentation. Please do feel very free to drop in questions into the chat, and then we'll engage with them after their presentations. Dr. Baso.
Thank you, everyone. Is my presentation visible?
It is. Please proceed.
Thank you. Thank you, Rob. So my presentation, I'll just giving a snapshot of what I propose research is about. So for this project, I want to see if we can build strong food webs for African freshwater ecosystems so that they are able to withstand the climate and invasive species that they are currently experiencing. Let me see. I don't know why it's not going through. Cool. Thank you. So the what of this project, as I've already said, we want to study how climate change and invasive species affect food web interactions in African freshwater ecosystems. And this will be done by quantifying the disruptions that happen due to these two factors. And we also look at how native species adapt to these changes, both in terms of climate change and invasive species. And then lastly, we'll explore the role of biological control in building a resilience within these affected ecosystems, especially with regards to invasive species. And so why would you want to do, or why do I specifically want to focus on the study? Mainly it's because we know that climate change is taking place, and we'll then want to know what the effects are in the ecosystem so that we can be preemptive in terms of protecting our ecosystems. And secondly, we want to know what the effect of invasion is, and what specifically for this project is the link between the two, and how both of these factors affect the trophic interactions or the food web within these ecosystems. And so what? So climate change would be down here affecting the primary producers, which is the plants and the lower level trophic levels. So what happens when we have an invader as the primary producer in those systems? That's one of my questions. And then what happens when we have increase in temperatures, increased rainfall events? What does that mean for the overall trophic chain in these ecosystems? And so where will this study be taking place? My targeted regions are listed on the screen here, but I understand that this could be a huge study for us. So we'll have a subset of these we'll choose from these countries for each of the objectives that we have. And so we can't do this alone, of course. So we have collaborators and partners in the different regions, with the SIEB being the host institution, which I'm based at, and the Center for Biological Control will provide the biocontrol agents and the expertise related to that. And then we have partners in the various regions as well, the University of Namibia, University of Botswana, University of Zimbabwe, Madagascar. We have partners in Lesotho. So we have a vast network, and it's possible for us to they will allow for this project to go smoothly in terms of the what do you call it? In terms of accessing the different sites and the government organizations that are relevant for this type of study. And then we also have international collaborators here, which will mainly help in building the different predictive models, and also building the study methods and the framework for that to make sure that it's rigorous enough. And then the how of this, I think I've already hinted to this. We'll have extensive field monitoring of the different invasive species and the native species in the different regions. But ideally, we want to have one or two common species, common invasive species across the different countries that we'll be working with, and then see how those invasions influence the trophic interactions therefore. And then the second part will be to simulate environmental change in lab experiments, whether it be temperature, nutrients, to see how that affects the trophic interactions as well. And then once we've done that, we can then build our predictive models to help forecast how invasion by specific species will affect the trophic food webs, and then help the managers or policymakers and managers predict and successfully manage invasions preemptively. And then lastly, this project will be in 3 phases, with the first year being mainly focused on research planning, and then for year 2, we'll be doing intensive field data collection and analysis. And then year 3 will be the final data analysis, reporting and dissemination to policymakers, the public, as well as the affected communities. Thank you very much.
Well done, and thank you for that presentation. So exciting research that is proposed, and I'll ask people to get their questions ready for a little bit of analysis. I'm going to ask you one very quick follow-up question. What is the core item that you hope to produce with this research? What is the output which will be valuable in the hands of policymakers that you just mentioned?
Thank you for that question, Rob. So the most valuable part for my research, I think, is I know it's very scientific. The predictive model will be the basis of everything that will come after that. Because even after I'm gone, the model can help policymakers and managers to predict what invasion will do to their ecosystems, and that then translates to protecting the ecosystems to make sure they are safe from the vulnerabilities of climate change. So we can have preemptive management strategies, which can then help to protect those vulnerable communities as well. And then I think that the other most important thing is to make sure science grows and is innovative, and is it's about building the next generation of researchers. I think that's the main thing that the Jennifer Ward Oppenheimer Research Grant will allow me to do, to have students that I will mentor to make sure that this research does not only end when I leave, it can also be incorporated into future research of the next cohort.
Fantastic. Great. Well, thanks very much. And now we're going to move on to Dr. Conradie.
Right, thank you. Yes, so as Robert briefly mentioned during the introduction, I am passionate and curious about the relationship between environments and animals and how they actually respond to the environment. And I'm interested in examining this through multiple different fields of discipline. So the project here is to use physics and biology to actually say something meaningful about how animals respond to environmental change, particularly in the context of global change. So when we talk about global change, it really does threaten biodiversity. And it threatens biodiversity in a number of ways. sorry, my screen is not advancing, which I'm not too sure why. Oh, there we go. Right, so yes, global change. What we're seeing is that environments are already changing. So this figure over here on the left-hand side shows the change in forest cover from 2000 to 2022. And what we can note is anything in yellow, orange, or red is habitat loss or forest loss. And anything in green is forest gain. And there's very little green on the map. So globally, we're seeing large areas where habitats are being lost. And then on the right-hand side, we have a map showing built-up land. So we're having lots of areas that are actually being transformed for natural areas being transformed for infrastructure and building. So this map over here is only in one direction. You're only seeing more built-up areas. You're not seeing less built-up areas. So we know that habitats are changing. And at the same time, it's predicted that temperatures are going to get hotter and the climate is going to change. We're going to have more extreme weather events, weather patterns. And what I'm interested in is looking at this combination. How does change in habitat structure and a warming world actually affect biology? And one of the things we know is that physiology and behavior really underpins species responses to global change. So you need to understand the biology and their physiology and behavior to actually address this question. So basically, I'm looking at understanding this complex relationship between animals and the environment, but I'm looking at a microhabitat level in a changing world. And what I mean by microhabitat, that is the environment that an animal is directly experiencing. And then I want to take this information and actually integrate it into evidence-based predictive models that can guide our conservation and management efforts. So just to go back to the microhabitat, what do I mean by this? Well, if you imagine that you're a bird sitting on a rock on a hot day, you'll be experiencing direct solar radiation. So you'll be getting quite hot. And if it is a hot day, your body temperature is going to be increasing. You might start panting to try and keep yourself cool. Because you're panting, you're actually losing a bit of water. So now you're at this risk. It's hot. My body temperature is increasing. I'm going to start panting. But if I pant too much and lose too much water, I'm actually at risk of dehydration. So you have this trade-off between conserving your body temperature or losing too much water. But if there's a tree available, you can go and fly up and sit in that tree and actually have your body temperature cool down, and then you're not really at that risk. But that means that that tree has to be available and that the temperatures in that tree need to be cool enough that you can mitigate this cost. So the first part of my project will be looking at, well, is the tree available? Let's start describing the natural environment that animals experience on a day-to-day basis. And then once we're able to describe that sort of thermal landscape, we can actually use what is called biophysical models. So those are models based on maths and physics to predict physiology in those environments. So you can predict the risks of dehydration and hypothermia. And then I'll also be looking at, behaviors. What does the habitat structure, how does that actually define whether or not species are breeding? And you know that species need to breed for populations to be stable. And then look at, well, how does that actually tie in to survival and vulnerability of species? So that's essentially what I want to do, but how do I want to do this? So I'm going to give it a little bit of a case study, sticking with the birds as an example. So this is a satellite image of an area in Kruger National Park. And you can see there's a river on the left-hand side, and all of those green dots are trees. Now, I aim to use things like the satellite imagery together with remote sensing to start categorizing the actual physical landscape. So this next figure over here, each one of those dots is one of these trees that we're seeing in the satellite image, but the color there corresponds to the height of the tree and the density. And when we're talking about density, we're basically talking about the shadiness. So how much shade are you getting in that tree? But that's the physical landscape. So what now can we say about biology, about species actually operating in that physical landscape? Well, we can actually say quite a bit. So like I mentioned, we have those models, biophysical models, where we can predict relationships of body temperature and hypothermia. I did that for a hornbill. So again, each one of these dots is one of those trees of different heights and different level of shadiness. But this time, those colors correspond to body temperature. And the closer it gets to red, the higher the body temperature of that bird would be in that tree, and the more thermal risk it would likely experience. But this is under current climates. I then looked at, well, what would that risk actually be under future climates? assuming that the habitat stays the same. So the same trees are there, but we just have a different or hotter climate. And then you can see a lot more to the red. So we're starting to see, okay, well, these are the thermal risks that birds might experience in these habitats, and that can start to inform how we manage these systems. We can see which trees are actually better, where they're not at as much risk, and identify those exact tree species and look at conserving those areas. So essentially, that's what my JWO grant application looks at. It looks at defining the landscape in terms of the physical landscape, but not just trees in Kruger National Park. This will actually be deserts through to forests, and also a freshwater component. So part of it will be actually modeling temperatures in freshwater systems and seeing how that actually affects communities. And then it will be to predict risks within these landscapes and to be able to produce maps like these across broad scales that start highlighting areas where species may be vulnerable. So over here, you can see there's a gray patch on the right-hand map there. It looks like it hasn't been colored in. That's actually an area where these birds are vulnerable and are not breeding. So you're starting to actually see, okay, well, these are areas we might need to actually look at conserving better. And I aim to do that for small mammals, birds, and reptiles. I just stuck with birds for the illustrative point of the presentation, but it will go for a range of organisms, with the ultimate idea of being able to provide guidelines on how to conserve and manage biodiversity in a changing world. And it's really important because having this tool will be able to highlight which species are vulnerable, as well as which habitats and areas are vulnerable and might need either conservation or management or both. And it's a really non-invasive, broad-scale approach that we can actually use to monitor biodiversity, because we're using remote sensing, we're creating mathematical models to predict this, and then we can actually shed some light on ecosystem health and resilience for biodiversity at a fairly large scale. So in a nutshell, that is what I aim to do, and thank you for listening.
Thanks, Shannon. That's really exciting work. I was interested in the bringing together of the very fine scale at a physiological or individual level with this remote sensing or satellite imagery that can be used to sort of characterize these landscapes. How much of the area, what area are you going to be focusing on? Are you planning to work on the methodology and approach so that it can be applied elsewhere, or does your study involve actually doing the analysis across large bits of the region?
Yes, so a bit of both. There will be the idea is to develop a tool that can be applicable globally, essentially, not just limited to a certain site. But then I will have certain sites where I actually ground truth the model predictions, right? Because we can't just have these great models and not actually know are they accurate or not. So there'll be sites in arid systems, so in the desert, some sites in more mesic forest environments, so places like KZN, areas like that, and then also Zambia, where it's more tropical climates. So a couple of case studies and then a model that is applicable broad scale.
Fantastic. Thanks very much. Again, all the audience, please do save your questions, pop them in the chat, and we'll certainly be getting to those soon when we come to engage with the three finalists. The third finalist is Steve Baleba, Dr. Steve Baleba. Welcome to this, and yeah, please share a little bit of background and insight about what you'll be exploring if you are to win a JWO grant.
Okay, cool. So, hi everyone. My name is Steve Baleba, and I'm a research scientist at the International Text Physiology and Ecology in Nairobi, Kenya. And the project that I have submitted to the JWO Grant Selection Committee is entitled Promoting Pollinator Diversity and Crop Yield Through Targeted Habitat Restoration in West African Agro-Ecosystem. So I am sure that each of us here has already encountered butterflies, bees, and overflies in our garden, or when having a walk in the nature. And what we usually don't know is that little insects called pollinator play a fundamental role in our environment. And for example, for us to have fruits like cucumber, apples, tomatoes, and grapes on our tables, these insects have to fertilize plant ovary with pollen. And if this fertilization process does not occur, we won't have enough cucumber, tomato, or cucumber, tomato for our burgers or salads. Also, we won't have enough grapes and apple make juice and wine. And unfortunately, this is what is gradually happening nowadays. And human activities such as pesticide use, the overuse of land, and climate change negatively affect the diversity and abundance of these beneficial insects. For example, this short video that you are watching here, this video was recorded by a farmer, by a bee producer who spoiled his colony after the neighbor had sprayed pesticide on their farms. So in the project that in my project, I aim to identify degraded site in East African countries and restore them by planting flowers that can attract diverse species. And through this action, I predict to improve crop yield in the farms neighboring the restored site. Also, I plan to train farmers on the use of natural method to control insects that are attacking their crops. And this method will include the use of parasitoid predators and fungus. And through this, I hope to reduce farmers' dependence on pesticide, and which are known to keep beneficial insects and contaminate environmental resources like soil and water. So at the end of the project, farmer will be equipped with sustainable agricultural practices knowledge that will help them to improve their crop yield and also at the same time to protect their environment. So with this, I would like to end with the presentation here, and I will be very happy to answer all your questions during the Q&A interaction. Thank you.
Thanks very much, Steve. Wonderful to get that vision for transforming those critical problems of pesticides and the destruction on pollinators. I'm really interested to find out a little bit more about your background and where you came from as a researcher to have this insight about what needed to be done.
Okay, so thanks. So I have a PhD in entomology insect science. So my background involved different areas of insect science, including behavior of chemical ecology, conservation, field trapping, biostatistics. So it's a bit, yeah, I'm an entomologist.
Fantastic. So great to meet you all. And I'm wondering if we can just turn on the other cameras and just spotlight the other finalists. It'll be really great to sort of get to know each of you a little bit better. And to do that, I want to ask a general question, which I'm going to ask to each of you specifically. And that's really about where did you get a sense that this was a direction that you were going to take, that this was really important to you. And I'm really looking to understand perhaps from where you started or why you felt like this was a critical issue to explore. So maybe I could start with Nompumelelo. Why are you doing this, and why do you get up in the morning and think about invasives in a changing world?
Mm-hmm. Thank you for that question, Rob. So the main reason for studying invasion specifically, I've seen a lot of vulnerable communities, specifically in Africa, struggle to access fresh water for fishing, for water sources, and for other uses, because the systems were so invaded by various invasive species. So that is the main reason why I wanted to target invasion specifically, and also to use biocontrol because that is been shown to be the most accessible way and environmentally friendly way to manage invasions. It's very accessible to local communities, unlike chemical pesticides or manual removal, which can be very labor intensive. So biocontrol is a good option for vulnerable communities specifically. And with regards to climate change, that's a topic on everyone's mind. So we can't. So we know that the system we're living in, the world we're living in, and the systems thereof are changing because of climate. So whether you're studying invasion, birds, agriculture, climate is always relevant. So that is why I am interested in that as well. But mainly it's because of the vulnerable communities that are impacted, and how do we find accessible solutions for those communities.
Fantastic. And I'm just listening to the conversation about biological control. Won't you, for the benefit of those who haven't perhaps even had direct experience of it or been involved with projects that apply it, can you give us a quick example or a quick perspective on biological control and why it's such a powerful tool?
Thank you again for that. So we know in South Africa we had, or let's say we have a variety of floating plants which are very problematic. Water ice, salvinia, parrot's feather. Those in the field will know what I'm talking about. But essentially these, they cover up entire ecosystems and they make it hard to actually access the water, as I've already said, for various ecosystem services. So we have had success in multiple occasions, specifically when it comes to the floating microphytes, Azolla folliculoides, Salvia molesta. We have biocontrol programs that are very successful for many of the floating microphytes that we have. So because of that, we find that some of these, so even though we have the technology and it's successful, but I think it's an access issue more than anything. So you want to make sure that everyone has access to this very exciting technology, and we don't have to rely on chemical spraying and manual removal, which can be very expensive.
Fantastic. Thanks for that. Just one more quick follow-up, and that is around the social aspects of this work. You've mentioned communities who are affected, etc. Does your research plan include social aspects, understanding how people living close to a particular site are interacting with that site? Or is it very much a biological study?
So primarily it is a biological study, but we can't exclude communities from that. And I'm aware that they might have important information to share about the ecosystems in which they are closest to. So part of the reason why we have those collaborators specifically in those regions is to have access to those communities and get that information about the system, get how they are affected by these challenges, and how they've seen the changes in their relevant ecosystems, and make sure that whatever solution we bring to them, they're part of the solution as well.
Fantastic. Thanks for that. So let me move on then to Shannon. Shannon, really to go back to that question around motivation and interest. Why and how did you come to this topic and this way forward?
Yeah, thanks. I've always sort of enjoyed animal watching, bird watching, animal watching, and just seeing how different animals operate differently in different environments, or how the same species might be doing different things in a different environment. So I've always kind of had that natural curiosity. But the idea of bringing habitat structure, climate together to understand how animals are responding kind of goes back to a moment. So I spent about 3 months in the Kalahari Desert 2-3 years back for field work. And it was long days in the hot sun, and I just finished a 17-hour day in the field. Heading back, it was a successful day, and I was sitting in the back of the bucky, you know, driving over the dunes, and it was like the perfect night, clear skies. And I just remember thinking, as far as I can see, it's just nothing. There's nothing there. It's kind of untouched, just natural habitat. And how many places in the world can we still have, or do we still have this experience today? And what is it going to look like in the future? Like in, you know, 50 years time, in 100 years time, in 200 years time, are we still going to have these natural environments where it's sort of untouched? And although the desert is such a harsh environment, and you know, midday you just want to get out of there, it's terrible. Night comes and there's this peace coming, and life is there. So it sounds a little bit silly, but that was a moment of almost clarity, being like, I really want to investigate habitat structure, climate, and be able to say something so that we can preserve this, so that we can still have these untouched natural landscapes that are broad scale. So yeah, I think that was kind of a moment where I was like, I'm going all in this kind of research, and that's what I want to do.
Oh, that's fascinating. Yeah, I hadn't thought about panting. I knew nothing about birds panting as a cooling mechanism. I was in the Kruger last year at the beginning of a WWF project, and we were just suddenly became aware, in fact, also with the hornbills, of these creatures sitting there totally still and just really obviously just struggling with that heat, and was really quite alarming. And I think in a previous conversation you were saying, you know, these animals, in particular in the desert regions, are very stretched anyway. They're at the limits of their capacity. So does desert environments then provide an opportunity that helps you learn a little bit more about this phenomenon?
Yeah, I would definitely say so. Like I said, because these environments are already harsh, so a lot of animals are already living sort of close to their limits. And by researching that and understanding that, you can almost use it as a glass crystal ball to see what other species might be doing should their environments become more hot, more extreme, more harsh. So there's definitely insight to be gained there, but also you know, species biology will be different in different systems. So just because the deserts are harsh and species are living close to their extremes doesn't mean that they don't have adaptations for that other species living in other systems might not have. So a bit of both, I would say.
Yeah, absolutely. Exciting to see you bringing together the physiological and the physics. Has that been a stretch for you?
It has a little bit. I can't say that in high school I thought that I would be doing physics once I was finished with it. But getting into it, it's been a stress, but it's a lot more accessible today. I find understanding it, studying it, and being able to bring it, and because the outputs that you get, you can link to biology. So that's where my passion lies. You can actually see the value of it, and you can see the value of bringing in not just physics, mathematics too, and struggling, like Nompumelelo said, struggling with a code or with a mathematical problem, and then suddenly it's solved and it makes sense and it makes biological sense, is pretty great. And I've come to sort of the school of thought that almost anything can be described with a mathematical equation. It just depends on finding the right terms to describe it. So I'm enjoying the process.
Fantastic. All right, I'm going to quickly move to Steve. Steve, you spoke about your entomology work. I wonder if you can tell us about the work that you did in your PhD. Just give us a little bit of background to that work.
Okay. During my PhD, I did PhD in entomology, as I said, but I was more focused on the insect behavior and chemical ecology. So what's chemical ecology? So what I did during my PhD was to see, to identify like the odor that a flies called stable flies it's a blood sucking insect, how that
Steve, I'm struggling to hear you.
identify a suitable oviposition site. So, and
Sorry, Steve, can you hear me okay?
yeah, but
We're struggling to hear you a little bit. I'm wondering if you want to turn off the camera for a moment, and then at least we can get your audio clearly. Great.
can you hear me now?
Yeah, that's significantly better. Thanks for that. So do you want to just take us through that little piece of work again?
Okay.
I'm just interested to hear a bit more. We didn't hear much.
So I was saying, for my PhD, I graduated in 2019. So basically, my PhD was to identify reposition from that a fly called stable flies used to identify a suitable oviposition site for their eggs, and to use those attractants to develop a kind of control tool. Like if you have the attractant, you associate it to a trapping device. Will it increase or enhance the catches of the flies? Yeah. Then after my PhD,
Carry on.
have you said something? Yeah.
Okay.
After my PhD, I went to Germany to continue as a postdoc researcher, where I run a project. Basically, it was affect the ability of insects to perceive odorant. So it was the continuation of kind of chemical ecology, but going a bit deeper in the neuron, like rearing flies. I was using different species of Drosophila, rearing them at different temperatures, then seeing how the developmental temperature is affecting their ability to perceive the odorant at the behavioral level, and going deeper at the neuronal level. So you may be wondering, why am I joining the conservation now? Is that as I was in Germany, I watched a lot of documentary and even reading some review, and the population, the diversity, and the abundance of pollinator in our continent is significantly reducing. And one factor that people are claiming is that in our continent, farmers, they rely too much on pesticides to control pests. And what farmers usually miss is that pesticides have a long-lasting effect in our environment, as well as killing the beneficial insects like bees. They also contaminate the crops and even some resources like water and soil. So, and also during my stay in Germany, I noticed that during summertime, you see flowers usually bloom. You have flowers everywhere, and we see a huge population of different kind of pollinator, like bees and bumblebees. And I thought that if I go back in Africa, can I combine like the culture of flowers and the use of biological control tool to mitigate the decline population of pollinator that we are currently having in our continent. So when I came back, I just came in April this April, and I saw the call. I said, let me write a proposal on that direction and see whether I can where it will go. So that's how I arrived here. Yeah.
Fantastic, Steve. And yeah, I can see that passion for preserving diversity among our insect populations is really shining through. So all of the best with that proposal. So we have got about 10 minutes left in this conversation, and there are a number of questions that were asked in the chat. I'm going to quickly ask Nompumelelo. There is a rather technical question about how the use of isotopes, could we elaborate on the use of isotopes for elucidating how trophic webs are altered by invasive aquatic plants? That's a technical question, and I want you to give it a very simplified answer, just so that everybody's everybody's clear on that key idea that you're going to be using there.
Thank you, Susie, for that question, and thank you, Rob. There are two main basic ones, stable isotopes that I use, is the carbon and the nitrogen. So the carbon is mainly to see which food source the insects or the organisms in that system are feeding on. So in my case, we'll compare invaded systems versus non-invaded systems, or is systems with and without biocontrol, so before biocontrol and after biocontrol. And we want to see if there's a shift in what the insects are feeding on. So we use carbon stable isotopes for that. And then we use nitrogen stable isotopes to see what level of the trophic level do the various organisms which we've sampled belong to. So that will be important information. And then we will also look at oxygen stable isotopes, and that will tell us about the specific environment, whether they are experiencing changes in temperature between the different regions, which ones are experiencing more extreme events. And then we'll also look at sulfur stable isotopes, and that will tell us more about the inputs that these systems are getting in terms of nutrient loading. So I guess that's the simplified version of it.
That's the simple version. So I'm glad you didn't give us the complex one, and thanks for keeping it simple. There's a quick follow-up. Will all your data collection be done in this specific season? And is it going to be done concurrently across sites?
So that's the plan. So the reason why we have those collaborators across the different regions, ideally we want to have the data collection taking place in summer in all the different regions, and want to maybe have 2 summer seasons, maybe, and 2 winter seasons that we sample across, and then we can have that seasonality incorporated into the model as well.
Great, fantastic. There's a question, Shannon, that was provided actually to you before the webinar. It was about saying, how will your project contribute towards human community climate resilience? So I think it'd be interesting to find out your thoughts on how understanding that microclimate adaptation ability or resilience ability, but how does that impact on human populations?
Yeah, I think in almost in 2 ways. So one way is by preserving biodiversity and having green spaces does impact human well-being. People tend to be happier in areas where there's more biodiversity, it's greener. So there's a bit of a link actually to just human well-being, but also by preserving these natural habitats and biodiversity, you're actually preserving key ecosystems processes. So things that can actually play a direct role in human well-being and livelihood. So for instance, having these systems where these ecological processes are maintained can buffer the extremes of high temperatures in the city, right? So there is actually a connection between lowering temperatures with having green space in it. And another way, it's probably not directly answering the question, but what the work will also be doing is, to a small extent, looking at livestock production as well, and how habitat structures and temperature influence livestock. And that has a direct link to communities and human populations that are maintaining the livestock and their livelihoods that are dependent on it. So it doesn't necessarily directly address, how humans will deal with heat, but it does indirectly by the maintenance of biodiversity and livestock production.
Fantastic, thank you. I'd like to, in the interests of time, with just 1 or 2 minutes left for us to wrap up, I'd like each of you to paint a little picture about where we get to if your research gets funded. So I realize that ideas about the future are always a little bit of a guess. They're a hope, they're a dream. But I'd like to ask each of you to paint that little dream for us. You've thought about it a lot. It's new to us. So what are you hoping the future will look like if your research gets funded? Let me start this one with Steve.
Okay. If this proposal is important that at the end, I hope that will provide a more argument or more information that will convince farmers to reduce their dependence to pesticide and adopt more sustainable agricultural practices, like such as the association of flowers with the crop productions. Yeah.
Fantastic, thank you. Let's go to Shannon.
Thanks. Yeah, I would like to see a product, I guess, of understanding vulnerability in terms of biodiversity, but also by having these maps that highlight vulnerable areas, and actually then speaking with policymakers and with people who are better than I am at explaining why this stuff is important, working together with them to start explaining to people why it's important to conserve biodiversity. So to have both this is what we know the risks are and where the risks are, and this is why we need to do something about it, and to develop an action plan of how to conserve and how to manage. And there's a target to keep 30% biodiversity conserved by 2030, to be able to contribute towards that and to be able to say, well, here's areas that we need to focus on and work together on action plans to be able to do that. Fantastic.
And final word from Nompumelelo.
So I'm going to use a real life example. So in the CBC, we know that there's an invasive taking place and taking over in Zimbabwe called Amazon frogbeard, and we are aware that we share the same waters, or I don't know. So whatever is in Zimbabwe has some way to get into South Africa. So that Amazon frogbeard could become our problem in future. And so how my model would then help us predict how the invasion by that species or any other species would affect our ecosystems and our native populations. So that is the gist of what I'm trying to do, to be able to forecast if we get an invasion from a specific species, given the biology of that species, how is it going to impact our native populations, and how do we then make sure that we build in those resistance in our communities, be it by biocontrol, be it by other means, to make sure that sensitivity is reduced from that invasion.
Fantastic. So with those closing words, I want to really express sincere thanks to Nompumelelo Baso, Shannon Conradie, and Steve Baleba for significant inputs and significant contributions to future sustainability in Africa. The one of you, as I said, will be funded, but we're hoping that you'll find support for your work in other contexts as well. The next OGRC, sorry, thanks also to all the participants in the audience. Thanks for really insightful questions. It was really great to get those perspectives from the contestants. The next OGRC Tipping Points webinar is scheduled for the 26th of September. There's going to be a focus on indigenous knowledge systems with medicinal plants and safeguarding food systems in rural areas across our region. So really, have we done enough? Are we doing enough to make use of indigenous knowledge systems to secure people, planet, and place? So please follow the link in the chat box to register for that. And then you're also able to sign up for the OGRC quarterly newsletter, a summary of this webinar and an article and adverts about future webinars, as well as really interesting topics and conversations get shared in that quarterly newsletter. So please do sign up for that. And then anyone who missed any part of this webinar, or indeed any other webinar, to please find the webinars on the OGRC YouTube channels. Once again, thank you to all of you for your participation. We look forward to welcoming everyone to the next Tipping Points webinar. And we are just thinking of all of you as the final round of deliberations takes place with the Jennifer Ward Oppenheimer Research Grant. And we look forward to meeting one of you at the Oppenheimer Research Conference. Thanks to everyone. And with that, let's close off this month's Tipping Points.